Thank you for the opportunity to present my work here at the ASM conference today. I'm super excited about this opportunity to present one of the newest methods that we very recently developed. So it's really like very new. And the main purpose of my talk today will be to introduce this method to you and show you and hopefully get you thinking about how you might be able to use it in your own research. So I will really talk a lot about what you can do with this method. And then I also show you a little bit of our validation data and some of the data that we have generated on how we have applied this method so far, although we haven't applied it to many systems yet, but I hope that is coming in the future. So my talk is about the ultrasensitive protein stable isotope probing to investigate the activity and substrate uptake in microbiomes. And so this method relies on metaproteomics as the basic method. And then, we developed it based on it. So I will give a brief introduction to metaproteomics, to standard metaproteomics. And after that, I will introduce a new stable isotope probing method that we build upon metaproteomics. So first, briefly about metaproteomics. Right, so of course, many of you will know that there are all these different Meta-OMICS methods to study the function and activity in microbiomes. And so, the question really is, where does metaproteomics fit in? So, we have initially, for example, we have amplicon sequencing, which gives us a good overview of taxonomic composition in a microbial community. And you can, for example, look at different conditions, sequence everything, and see how the community composition changes. And then we have, for example, metagenomics and metatranscriptomics, which give us a good insight into the functional potential in microbial communities. But really, when it comes to actual phenotypes on a molecular level, we have metaproteomics and metabolomics that really provide us with insights onto which genes are actually expressed and active and which metabolic end products, for example, are being produced. So metaproteomics really is at this interface of looking at the phenotype on a molecular level for microbial communities. So, and just to give you a quick introduction to a general workflow of metaproteomics, of course, there's many different ways on how you can do metaproteomics. But so ultimately, all of them would start with the sample collection. You would collect samples from your most favorite environment and then bring them back to the lab, do a sample preparation method. And there's, again, many different types of ways on how you can prepare your samples for metaproteomics. Usually, you have to do some kind of cell lysis. You get a protein mixture, which you then clean up. And then ultimately, you digest the proteins into peptides, and you get a cleaned up peptide mixture, which will then serve as the input for the next step. A question that often comes up in the context of the sample preparation is, of course, how much material do I actually need to do a metaproteomics experiment? And so what I like to say, of course, it depends on your sample type, but, ultimately, if you have 0.5 milligram of wet weight material, you can actually start thinking about doing metaproteomics. So to get started on that. Okay, so you have this cleaned up peptide mixture. The next step then is to actually separate the peptides using liquid chromatography, and you can do one-dimensional, two-dimensional separation. Some of them are very long if you have a very complex sample. The eluting peptides are then injected into a high-resolution mass spectrometer, and hundreds of thousands of spectra are generated for your sample. And we then use this mass spectrometry data in combination with protein sequence databases to identify and quantify proteins. And for some samples, we can identify and quantify more than 10,000 proteins and tens of thousands of peptides, of course. And so once you have this data, then you can, of course, use it to really start understanding what is going on in your community. So you can use it to reconstruct the metabolism, physiology, and interactions within your community. Just to give you a few quick examples about the type of questions that you can address with metaproteomics. Here, this quick overview slide. So recently, for example, we developed metaproteomics into a tool that allows us to look at community composition. And you may say, oh, well, there are already a lot of methods to do that, right? So there's a lot of methods that allow us to look at taxonomic community composition in terms of counts, how many cells of each species or how many genomes of each species. So for example, 16S RNA amplicon sequencing would be one of these methods. Metaproteomics now allows us to actually look at the community composition, but from a perspective of mass contributions of individual species. And you can easily imagine that if you have cells of very different sizes, biomass of community members and counts of community members may be very different. Another type of questions that you can address with metaproteomics, and that's more the classical metaproteomics experiment, is really what is the metabolism and physiology of your community, of your species in your community. And so here, just to give you a quick example of how we have used this in the past, here we're looking at this gutless marine worm. It's special because it doesn't have a digestive system. It lives in marine sediments, and it has a variety of different symbionts that live under its outer skin. And so these symbionts are responsible for autotrophically fixing carbon dioxide and ultimately feeding the host. And for a long time, it was unknown how these, what energy source these symbionts use to actually fix carbon and provide nutrition to their host. And so there was a metagenomic study that came up with a lot of really good hypotheses, but ultimately it was metaproteomics that allows us to really identify one of the energy sources. So we found that in some of these symbionts, we have very abundant carbon monoxide dehydrogenases. And so these carbon monoxide dehydrogenases allow the symbionts to use carbon monoxide as the energy source to fix carbon. And then, of course, after metaproteomics allowed us to generate this hypothesis, we then followed up with other methods. We did physiological incubations and measured in situ. And so metaproteomics here was really the key to focus our perspective on what is going on in the environment. And then, of course, you can also look at microbial species interactions, particularly if you have a system that you can somehow manipulate different environments or artificially induce some environmental change, then you really can start teasing apart interactions in these communities. Another method, based on metaproteomics that we recently developed, actually allows us to look at carbon substrates of microbes in their respective environment. And so if you're interested in that, you can have a look at this publication. Just a quick self-advertisement here. Last year I wrote a perspectives article that really just gives a brief overview of the types of questions that we can address with standard metaproteomics, and it's just some basic resources of where you can get started. Okay, so metaproteomics is a wonderful tool, but of course there are limitations. There are certain questions that we cannot currently address with metaproteomics. And this is where this talk has come in, where we'll now have new approaches, advanced metaproteomic approaches that allow us to overcome some of these limitations. And so I'm going to introduce two of these limitations and then later demonstrate on how we can use the new approach to overcome these limitations. So the first limitation, of course, is the annotation issue. So many of you may be familiar that, if you, get your nice metagenome or metaproteome from the environment, you get all these different conditions or samples or substrates, you get all these expressed proteins in the different samples, and you see really important differences in expression. But then when you look at the protein annotations, you see that for some of them, there's actually a known function, but for many of them, there's no known, they are of unknown function. No known function has been discovered, so you really don't know what they're doing. So you may, based on these annotations, you may think that a certain substrate in your community is taken up by this organism based on a known function of a protein. But what you may be missing is actually that there are these proteins of unknown function that actually allow a different organism to also take this up this substrate, and maybe even better than the organism for which you have the known function protein. So the question really is here, how can we obtain direct evidence about who the actual main consumers of a substrate are in a microbiome? The second limitation is that protein abundance changes often can indicate actually activity changes, but it does not always have to be true that abundant proteins indicate high activity, even though oftentimes we can infer it. So for example, if you have changes that increase metabolic rates simply based on enzymatic regulation, that would not show up in a metaproteome. Or if you have organisms that are abundant but are currently dormant or may have died, then of course, they would not provide activity in the community but still show up as abundant in your proteomes potentially. And then a newer subject is that we also have to, of course, consider relic protein. In many environments, we have a lot of protein biomass from either dead cells hanging around or just that was secreted previously. But of course, this particular protein, even though it may still be active, it will not provide activities to any of the cells microbial species in your environment. And so the question here is, how can we obtain direct evidence of metabolic and translational activity and changes thereof, using advanced metaproteomics methods? So as I said, we can overcome these limitations now using some information that I would call hidden information in metaproteomics data that we can use now to solve this. And so I'll briefly introduce some underlying mass spectrometry data now to really look at what this hidden information really is, or in this case, it could also be called underused information. So what we see here is a mass spectrum, one of these hundreds of thousands of mass spectra that we acquire in a metaproteomic experiment, and each of the peaks corresponds to a peptide in your sample, right? And so, for example, we would know from identification of these peaks that we have a peptide sequence here represented as an amino acid sequence, and we know from which organism it is because we identified it. And if we have a good genome, we can relate the peptides back to the organisms. So now, if we look at one of these peptides in more detail within the spectrum, it actually reveals that there is more than just one mass peak for each peptide present in a mass spectrum. And so the reason for this is, of course, that in nature, most elements occur in multiple isotopic forms. So for example, for carbon, we have carbon-12, which is the most abundant isotope, roughly 99% of all carbon. And we have carbon-13, which is roughly 1% of all carbon on Earth. And so what you can imagine happening now is we have these peptides and they have hundreds of different atoms in them. And so this first peak is when all the atoms in the peptide are actually of the most dominant form. And then what happens, of course, with very high likelihood is that some of the atoms in a peptide are actually substituted by one of the heavier forms. So you will get another isotopic peak where one of the atoms is exchanged for the heavier one, and then two of the atoms, and so on and so forth. So looking at this pattern, we could actually look at the intensities of the peaks and say something about the isotope content of the peptides. And so just to illustrate this a little more, if we said we artificially add label carbon-13, the heavy form of carbon, to peptides, either 1% or 10%, that really would significantly change the isotope pattern for this particular peptide. So here in this example, we have 1% added carbon-13, and you can see that would overall decrease the intensity, so it's shown in red, decrease the intensity of these front peaks here in the isotope pattern, whereas then the later peaks would actually increase in intensity because there's more heavy carbon present. Now, if we added 10% of carbon-13, this would change even more. More of the front peaks would lose intensity, because of the heavier carbon, and there would be additional peaks that would show up in our spectrum because we have now more and more heavy peptide forms. And so what we did is we, together with our collaborator Mark Strauss at the University of Calgary, developed a tool which is called CALIST that allows us to very accurately measure the isotope content of peptides in our mass spectra. So we can use this tool to determine the isotope content of peptides, proteins, and ultimately also species. And so what can we use this for now, to actually coming back to the limitations I spoke about in metaproteomics? So what we can do is so-called protein-based stable isotope probing, or in short, protein SIP. And I'm going to briefly illustrate now a workflow, what a protein SIP workflow would look like, to make it clear what we can do. So what we can do is we can use a substrate that has artificially a higher amount of the heavy isotope. And so you can basically purchase these substrates, for example, carbon-13 CO2 or heavy ammonia or polymers or specific sugars. Almost anything can either be purchased or synthesized to order. So you can really think of all kinds of different substrates that might be relevant in your particular microbiomes. So you have this heavy isotope. Then, of course, you have to do some kind of an incubation experiment where you apply the labeled substrate to your community. We then run a standard metaproteomic sample preparation and LC-MS measurements. And we can, of course, then use this data for the standard metaproteomics workflow that I mentioned earlier. You can use it for reconstruction of microbial community metabolism. But at the same time, we can use the same exact data to now also process with our CALISP software to determine which proteins were labeled using this particular substrate and which species have been labeled. So we really can, make the connection between substrates and species and proteins. And so what can we ultimately do with this, right? So when we look at proteins, there's actually 5 different elements that usually occur in proteins, right? We have carbon, hydrogen, nitrogen, oxygen, and sulfur, and for all of which we have heavy isotope form. So all of them in theory can be labeled in a substrate somehow. And so what we can do, of course, we can now identify and quantify substrate assimilation by microbial species in microbiomes. So we can make the connection between substrates and the microbial species using them. And in addition, we can also use protein SIP to identify and quantify activity of species in microbial communities. And so that's because we can, of course, use heavy water. So we can use water, which is partakes in almost all metabolic and physiological reactions, and therefore is a really good marker of activity. And so we can label the water either with oxygen 18 or deuterium, so we can really use this as a marker for finding activity. Okay, so and now I'm going to briefly talk about a few of the validation studies that we did, and then also give some examples of the applications we have used this for. So we did initially, of course, a lot of experiments with pure cultures just to validate our workflow. And of course, I'm only showing just one of these many experiments that we have done. So in this particular experiment, we have Escherichia coli, which we grew in a defined medium with glucose. And what we did is we replaced part of the glucose in a medium in different incubations with carbon-13 glucose. So we added a heavy form of glucose, but in different amounts of the total glucose. And so now this is the data that we generated with this experiment. On the x-axis, you can see the substrate carbon-13 to carbon-12 ratio, which we added to the experiment. On the y-axis, you can see the same ratios, but measured with our protein SIP method. And you can see we get a beautiful linear relationship between the two. You may also notice that actually our measured quantities actually start roughly at 1%. And that is, of course, due to the fact that in nature we have already 1% of all carbon being carbon-13. So anything that we add as a label is on top of the naturally occurring carbon-13. So now what we found is actually that we can actually measure as little as 0.01% of added label incorporation into the proteins. And we can also see that this is fully quantitative. We get really nice linear relationships. Okay, so the next type of experiments that we did is we actually wanted to increase complexity and see if this particular approach also works in much more complex microbial communities. So what we did is we generated mock microbial communities in which we added 32 different species that were all not labeled. And to this mock community, we then added Escherichia coli that was grown in 5% carbon-13 glucose. So that really allowed us to see then if we can detect these Escherichia coli proteins within this mock community as labeled, and also we can see if this causes any kind of interference between the labeled and the unlabeled cells in the mass spectra, which could lead to false positive detection of label incorporation. And so to this mixture, we then again applied protein stable isotope probing. And now this is the data from this particular experiment. And of course, we again did many experiments with many mock communities. The main point I want to make here in this particular slide is that we are actually, in this slide, we're looking at incorporation into individual proteins. So as I mentioned, we can measure not only incorporation into species, but also into individual proteins. So here in this box plot, you see the carbon-13 to carbon-12 ratios of individual proteins. So here in this yellow, we have the proteins from the unlabeled mock community species, and in blue, we have the proteins from Escherichia coli, which all show some amount of label. And a lot of proteins, we can only detect with a single peptide, but for other proteins that have, are more abundant, we often get multiple peptides. And so, of course, one thing that we're seeing, if we have fewer peptides for the detection of a protein, there is a little bit of a false positive rate that we see where some of the unlabeled mock community species proteins actually show some kind of a label, so there's a little bit of a wobble. But the more peptides we have for the identification of a protein, the better this gets and the much lower the false positive detection of label incorporation is. However, even with as little as one peptide, we get mostly a really nice identification quantification of label incorporation. Okay, so now coming to actually a little bit of a case study where we now demonstrated how we can apply this to different kinds of questions. And so before I can go into our case study, I have to briefly introduce a small piece of literature here, which is from this science study from 2011. And so what the authors did in this particular study, they fed mice diets with increasing amounts of protein in their diet. And then on the y-axis, we see that we have increases of abundances of particular microbial species that they measured in this experiment. And so as you can see, increasing amount of protein in the diet leads to overall increases in microbial species abundances. And also, of course, the individual species become more abundant. But so the question that I'm asking here now is, does this higher abundance of species necessarily reflect a higher activity? So it's really an association between abundance and activity of microbial species. Or do these species grow up to this higher density, but then reach a steady state in the activity, the overall activity of the population remains then the same? And so what we did now to an experiment that looks kind of at this question is we got some data from our collaborators Nico Jemlich and Robert Starke, who had data from an experiment with bioreactors. And so what they did in these bioreactors, they had 63 isolated species from a gut microbiome that they grew together in these bioreactors. And these bioreactors were either fed with high fiber simulating medium, high fiber diet simulating medium, or with a high protein diet simulating medium. So these, were separate bioreactors. And so what they did, they subsampled from these reactors into multiple bottles each and added to these bottles heavy water with oxygen 18, and then continue to incubate to really use the heavy water as a tracer of activity. And so I will not show you all of the data that came out of this experiment because it's kind of an overwhelming graph, but I can tell you that we quantified the activity for more than 20 species in these microbiomes. So again, coming back to this literature data, right? Just looking at two of the species in this data set, B theta increase in abundance and Clostridium symbiosum increase in abundance. And now again, the question was, do they actually increase in terms of activity or just in terms of abundance? And how does that connect? So here's just a small snippet of the data that we generated with the bioreactor experiment. Here we have the high fiber medium and the high protein medium. On the y-axis, we are showing now the ratio of oxygen 18 versus oxygen 16, which is really an indicator of how much of the heavy water was incorporated and really is an indicator of the cellular activity. And so what we can see now here on this data really nicely is that actually B theta in the high protein medium really has a massive increase in terms of activity, which really nicely corresponds with its abundance increase in the mice. For Clostridium symbiosum, however, which is also increased here in the mice, we do not see a corresponding increase in terms of activity. It actually almost seems as if in the high protein medium, the activity decreases slightly. So overall, this really indicates that an abundance change does not necessarily always indicate a long-term increase in activity, but of course we have to still work out better what is really going on here. But this just illustrates that we can really use this heavy water as an indicator of activity in microbial communities. Another example that you could, for example, think about is if you, for example, have a permafrost soil that is now thawing due to global climate change. Microbes may be becoming active again, but they might not immediately start replicating or producing more protein, but you would immediately be able to track and detect activity increases in such a setting using heavy water, because you would see at least small turnover of proteins, which would lead to incorporation of heavy water. Okay, another and final experiment that I wanted to briefly show is an experiment with mice. In this case, we had mice. We produced a diet with fully defined components, and one of the components was sucrose. And what we did, we had a control diet with just all normal ingredients, and then we had one diet in which we replaced just 1% of the total sucrose with carbon-13 labeled sucrose, and we fed this to mice. And so now what we can see here in this plot, we see just some of the microbiota species that we detected. And you can see in the control diet, these microbes have roughly the same carbon isotope ratio. But some of the species, actually, when we feed this diet that has a little bit of carbon-13 in the sucrose, we see an increase in their carbon-13 content, which really indicates that these species must be consuming some of the sucrose in the diet, whereas others evidently do not directly consume sucrose. And with that, I would like to sum up my talk. I hope I was able to share some of the excitement about this new method that really should allow us to do all kinds of different experiments in microbial ecology. So using ProteinSIP, what we can do now, we can address questions such as which microbial species use a specific substrate, and how is it handed off to other community members? The latter part, of course, if you start thinking about pulse chase experiments or time series experiments, how does the activity of microbial species change in response to environmental change or between environment? Of course, we can use this heavy water protein stable isotope probing. And ultimately, as I also briefly showed us, we can detect incorporation into individual proteins. So that allows us to really see which metabolic pathways and molecular mechanisms are used, activated and deactivated in response to environmental or generally changing conditions. And with that, I would like to thank my wonderful team here at NC State, specifically Marlene Jensen, Angie Mordant, Fernanda Salvato, and Jessie Mayo, who were involved with this research, some of my collaborators here at NC State, our collaborators at the University of Calgary, particularly want to highlight here Angela Kouris, who did a lot of the heavy lifting in all the experiments that we ran for validation, and then, of course, all our funding sources. Thank you very much.